What a wonderful synopsis by all of our teams on the projects they've been so immersed in this summer. Thank you so much for those terrific presentations. Let's give ourselves another round of applause, please. You'll be hearing from 3 speakers now. They are the proud graduate mentors of the undergraduate students you just heard from a moment ago. We are going to start with Scott Smith. Scott Smith is a PhD student in the Department of Plant and Microbial Biology at North Carolina State University under the advisement of Dr. Zhongpu Shi. Scott will be focusing on identification of mixed linkage beta-glucan quantitative trait loci in hexaploid oat.
Okay, thank you for that introduction. So my name's Scott Smith, and I'm really excited about the opportunity I have to share some of our research with you today. So our focus has been on identifying some of these traits that my interns have talked to you about. But first, again, the Plant Pathway Elucidation Project, its goals are to define what these crops we're researching make, how they make it, and how that impacts human health. I have spent most of my time in the second area, which is how do these plants make these compounds. We know that oat makes a lot of good compounds that are good for our health, and we're trying to identify the process of making these compounds. So oat, what does it make? We know oat makes fiber. That's one thing that is obvious with our grains, is they make a lot of fiber, and we always assume that is most likely the reason that oat offers so much health benefits to us. But oat also does have a lot of other phytochemicals, a lot of phenolic compounds, which also contribute to these health benefits. Typically, when you hear about antioxidants and anti-inflammatories and things like that, you often think of your fruits and your vegetables, and that's because in these crops, those phenolics are usually in their free form, and so they're easily accessible, easily absorbed into the bloodstream, and so they've been studied a lot closer, where in oats and our other whole grains, those are in bound forms, and they're not as easily accessible, even though our bodies through digestion can access those. They haven't been as easy to study, and so it's not typically what you first think of when you think of oats. You typically first think of fiber, but they do have a lot of phytochemicals that are very good for our health. Some of these compounds that we're interested in is beta-glucan. This is a fiber, and it is the reason that General Mills can put this health claim on their box that Cheerios can help reduce cholesterol, and that's because of beta-glucan. And also, oats have avenanthramides, which are very unique to oats. And they also have tocopherols and tocotrienols, which are vitamin E's. And these provide anti-inflammatory, antioxidant properties, which provide health benefits to us as well. So a lot of time has been spent in this program to develop the genetic resources that are needed to that they're needed to answer some of these questions. What do these plants make? How do they make them? And how do they benefit human health? So the first couple years of this program, I have worked on developing an oat consensus map, which will give us the information we need to link these traits to the genome. So our oat consensus map, it was developed using 12 different populations of oat. And this is a figure that represents the map. Typically, maps haven't been as high density as our current oat map is. And so this graph here depicts the density of markers along the chromosomes. And it's a very high density map, which is essential for us to make these associations. And the oat genome has been something that's very complicated to work with. It's a large genome with about 11.3 gigabases, and it's a hexaploid, and so it's very complex in nature, which makes it difficult to work with. And one of our goals also is to sequence the oat genome, which you've heard the interns talk about a little bit, and that is a very large challenge. And once we have that genome sequenced, we'll have a much better resource to help answer some of these questions. But now that we have the oat map in place, we can start looking at some of these questions. And the way we want to look at these questions, we want to link these phenotypes, beta-glucan, avenanthromide, tocopherols. We want to link those traits to the genetics. And we use that doing a genome-wide association study, which links those genotypes to phenotypes. For us, we have 3 study populations that we worked with. Our first population was a population of spring ecotypes, and these were lines in North America. The winter population is, again, North American lines that are winter ecotypes, and then we also had a diversity panel, and that represented oat lines from across the world. And all these populations were grown in 3 different locations, Lacombe, Fargo, and Aberdeen. And we were able to get phenotype and genotype data for all 3 populations in 3 or 2 of these locations for each of those phenotypes. And having that data helps us to make these associations, and we would really like to thank General Mills for getting that data for us. So now that we have this data, we wanted to check this data to make sure that we make sure we cover our bases and make sure that our genome-wide association studies are effective and that we account for all the different things we need to account for. So we did a lot of pre-analysis of the data. One of those is to look at the population structure of these different populations. So we first combined all of our spring and our winter data together into one data set. And when we looked at that through principal component analysis, we were able to see that these 2 populations are very different. You can see on the left graph here, the spring populations all cluster together, and the winter populations all cluster together, and they're very distinct from each other. And so we knew that these populations are very different from each other, and we knew that would bring bias into our association study. And so we decided to treat these 2 populations independent from each other and run our genome-wide association studies on these populations independently. And when we did that, we also again did principal component analysis and kinship analysis to look at the structure of these populations so that we can make sure we account for that structure in our analysis so we don't have any false positives due to our population structure. And the graphs on the right, they show one is from our spring population, one from our winter population, and they're colored by breeding programs, and we can see that they tend to cluster based on which breeding program these samples came from. And so we know that a lot of the structure in our population is due to the breeding that has been taking place with these different breeding programs. And so now that we know that, we can account for that population structure and reduce the amount of false positives we have in our data. Next, we wanted to look at our markers in our study to make sure that the markers we're using are informative and that we have markers that are going to be helpful in making these associations. So we did what's called a polymorphic information content analysis, and this shows us on this graph we have our linkage groups here at the bottom from our consensus map, and on the right hand side it's the position, the centimorgan position on the linkage group, and each dot represents a marker, and it's colored blue to red. So dots on the map that are red are markers that have high levels of polymorphism between the individuals in the population, where blue ones have very little differences in alleles. within the population. And so we would hope to see a lot of red because that means that there's a lot of variation in our population for those markers in these populations, which would make it so it'll be more likely to find associations, and if we find associations, then there'll be variation that we can increase or decrease the levels of these compounds. Next, we also wanted to look at the linkage of these markers. And this is a linkage disequilibrium plot where on the bottom we have genetic distance, and on the right we have the degree of linkage, and each dot on the plot represents a comparison of two markers on the same linkage group. And so you'd expect that markers that are close together on a linkage map would be linked tightly together, where markers that are farther apart on a linkage map would have very little linkage between them. And so as you get further away, you see that the level of linkage decreases. And that's what we would expect to see in a linkage disequilibrium plot. And we plotted a spline to this that just shows the average linkage disequilibrium across all the chromosomes for all the markers. And then typically 0.2 is where we would say that markers become unlinked, and that happens at a distance of about 5 centimorgans. And so this tells us that our linkage between markers decays at about 5 centimorgans, which means for our associations to be effective, we have to have a marker density in our map of at least 10 centimorgans, or one marker every 10 centimorgans in order to make effective associations. And so we went back and looked at our map to make sure that our map was dense enough to use in a genome-wide association study. And on average, our density is 0.78 centimorgans per marker, so that's a lot greater than 1 every 10 centimorgans. So we have definitely enough density in our map, and we had very few gaps that were larger than 10 centimorgans. There are only 10 gaps in our map that were greater than 10 centimorgans. And so now we know that our map is going to be an effective map for our genome-wide association studies. And we've accounted for our population structure, and so now we can go ahead and run our genome-wide association studies. And so, like I said, we had 3 different populations and 3 different locations, and we had avenanthromide, tocopherol, and beta-glucan data to run all this genome-wide association studies for. And so that's a lot of studies to run. And so thankfully we had all these interns that each intern could get on a computer and run one analysis, and so we were able to pump through these analyses really fast. I'm not going to show you all the results because it's a lot of information, and they have them on their posters, and I want to take away from them sharing their results as well. But I'm going to focus here on some of the beta-glucan data, some of those results. And here we have a QTL plot for our winter population, where again we have linkage groups on the bottom, and each linkage group has the 3 different locations that they were planted in. And because we're running a genome-wide association study, we know that just the nature of these studies, we know there's going to be a lot of false positives with our markers that we find that are significant. And so what we did to control for that is we considered highly significant loci as ones that were replicated across locations. And so we can see that we have our linkage groups here and our position on the y-axis. So those represent our linkage groups, our chromosomes, and these highlighted dots are the ones that are significant. Anything that's gray is insignificant. And these ones that are boxed are areas where we have markers that were replicated across at least 2 of our 3 locations. And so here we have replication of markers, same marker that was significant in multiple locations. And so we can see from in our winter population, we had 5 QTLs, which are these 5 regions with replicated markers. And then for our diversity panel, there were 10, which are these boxed ones. And then for our spring, we had 10 as well. And from all of these combined, we wanted to narrow it down to ones that we really wanted to focus on when we go to the next step and look for candidate genes. And so we wanted to focus on these three. And the reason we want to focus on these three is because there's multiple markers at these positions replicated across the locations, and they're also replicated across studies. So this position was replicated in both the spring and winter populations, and this one was replicated in spring and winter, and this one was replicated in spring and diversity populations. And so these give us a lot more confidence that these QTLs are going to be real QTLs with genes that control these functions, or these traits, beta-glucan. And so these are the areas that we want to first look at to see what genes we can find in those regions to see what's controlling these traits. So our next step, which we haven't gotten to yet, is to take those significant markers in those QTL regions and get those sequences associated with those markers and see where they fall in closely related species like barley, brachypodium, and rice. Again, we don't yet have the oat genome sequence. If we had the genome sequence, we could go directly to the oat sequence and see what genes are there, but we don't have that resource yet, and so we have to depend on related species. And so we'll look into barley, rice, and brachypodium and see what genes are there, because those have been sequenced, they've been annotated, and if we can find these regions of microsynteny with our QTLs and these other genomes, then we can be confident that any candidate gene that we find in these regions, as we look through these genes here, if we find some that look like they could be involved in this, we can be confident that that same gene is going to exist in the oat genome at that same position. And so that's our future work. And once we identify these genes, the next step would be to test them and to see if they actually are involved in the pathway. And we do that using transgenic approaches in either Arabidopsis or in rice, depending on what the genes are. And then we can test them and see whether or not they actually are involved in the pathway. So that's our plan going forward. And I would like to thank my interns who helped me a ton this summer. Without them, I wouldn't have been able to get nearly as far as I have. I'd also like to thank General Mills, who provided us with this data, and also for my advisor, Zhang Fu, who's given us a lot of help and direction as well. And if you have any questions, I'd be happy to take some questions.
Thank you, Scott. Let's give it up for Scott and Team Scott. So I said at the beginning that I'm Kim Parati. I think you should know I'm with WFAE. I'm with WFAE, the NPR news talk station, and my main job is to do corporate development, where I do limited research, more demographic kinds of studies. So I'm completely amazed by all of the work that's going on here at the North Carolina Research Campus. I'd also like to go ahead and say, I should have started off with this, I apologize for any butchering of names. Please don't kill me. You can get me after during the break. So the next graduate student we're going to hear from is Weston Busler. Weston is a PhD student in nutrition at North Carolina State University under the advisement of Dr. Slavko Kremenitsky. He earned his BS in nutritional studies at the University of Minnesota Twin Cities, and his research focuses on connecting food development to promote human nutrition. Wes's piece will be on optimizing human health with plant genomics. Thanks, Wes.
Thank you. Make sure I get everything together here. Okay. Okay, here we go. Well, once again, I'm Weston Busler, PhD student in nutrition at NC State University. Today, I'm going to be talking about how we connect some of these plant genomics with human health aspects. And a lot of what we've been talking about so far has been just looking at the nutrients, and what I've been really focusing on is the actual nutrition effects. So, First, we want to focus on the known health benefits or health associations that we see with these plants, that's been seen prior with these plants and diseases. So, heart disease. Heart disease is the #1 killer of Americans, and it's been so that way for quite a long time. Now, what's been found is that consuming cruciferous vegetables, such as broccoli, will significantly decrease the amount of disease that you see, as well as the progression of the disease. So you can prolong life and prevent some of these morbidities. Cancer. Cancer is the #1 or the #2 killer, and specifically with broccoli, you've seen that an increased consumption has decreased the incidence of certain types of cancers, most notably colorectal cancer. Broccoli. Broccoli is a nutrition powerhouse. On the graph behind me, there is the dietary reference intakes as a percentage of the dietary reference intakes. Each bar shows that all these metabolites have different amounts in broccoli, but they're all based on averages. So when what we're looking for is more of the outliers within these averages and what makes some of them more effective. And specifically, we're looking at the antioxidant components in broccoli. So vitamin A, vitamin C, vitamin E, flavonoids, and glucosinolates. These are the primary components of broccoli that have been associated to the antioxidant potential. All right, then what we need to find is, are these metabolites biologically relevant? The biggest thing when you consume a food is that it has to go through metabolism. And in this, some things get through, some things don't. Some things only get the benefit, like in lower gastrointestinal fermentation, so not directly from your initial first-pass metabolism. Then, what we need to know is what nutrients do most of the work. Also, does a mixture of these certain nutrients help better? So, in plants, there are certain factors that contribute to this variation. You have the genotype of the plant, so what genes are causing what, and they're making it express different levels of certain metabolites. The environment that they're grown in, because people are a great indication, the environment that you're grown in really affects how everything goes, like how tall you get can even be affected, the size that you get, there are all sorts of things. Also, with plants is the processing conditions. Once the plants are picked, how they're prepared, how long they're stored, whether they're frozen or not. These all contribute to the nutritional properties. For P2EP in this summer, or the past 2 summers, I might add, the genotype has been where we're focusing. So in plants, we've been looking at, or in broccoli specifically, the anti-inflammatory effect. And as my interns went over earlier, there's a specific reason that these inflammation markers help in reducing some of these chronic disease risks. So the graph behind me shows a line trend as well as bar graphs. The bars represent cell viability. Now, when you treat with broccoli extracts, one of the big things is you want to make sure that your extracts aren't actually killing the cells, and we're treating these in immune cells. So these are the same kind of cells that your body has, and when they respond to infection, they engulf whatever it is they're trying to get rid of and destroy it violently. But then you want to quench that as quick as possible. So the leftovers are the free radicals. And when we do this in a cell model, we want to see the amount of extract that is reducing the nitric oxide, which is the free radical measurement in this case. So the trend line that you see shows at 30 micrograms per milliliter, broccoli extracts were reducing nitric oxide at about 50 to 60%. So this is a pretty significant effect, and it is not at all killing the cells. So that shows that it's a true relation. So then, how are we going to map these health benefits to the plant genome? What we're going to do is conduct a nitric oxide experiment on a mapping population of broccoli. And this allows us to do a QTL study, some of which has been described earlier, but it will use the genetic resources from that specific mapping population, the genetic linkage map that's already been produced, and then we're going to be able to assign values to each of the genotypes. So for this, we used a F2, F3 broccoli population from 2 distinct parents. through them, we were able to get 150 unique or slightly different isogenic lines, which means they're almost the same, but slight genetic variation from the 2 years. And then, so then we did the study in 2 years. And this allowed us to get more reliable data by growing the same thing under 2 years, since there are all those environmental conditions that can happen. So with this, we did a 30 microgram per milliliter treatment, just as the other study did. And we measured dissolved broccoli extracts in dimethyl sulfoxide, which was used to treat macrophage cells that were stimulated with lipopolysaccharides, which are the outer layer of gram-negative bacteria that are often associated with diseases. From this, we wait after the treatment 24 hours, and we use a grease reaction to calculate the amount of nitric oxide remaining after this incubation. So what we can do is, through a colorimetric assay, see the changes in free radicals in all of these different cells. So from there, we can look at the differences between the controls and our new treatments and have a % change that we can use as our primary value. What do we see? It's that not all broccoli is created the same. So this is the scope of our entire population study that we did, and some of the samples were performing extraordinarily better than others. And this is a good thing for us. It allows us to look at the variation to a much stronger extent when we go to our QTL study. This is the QTL study output. The long dotted graphs are the genetic linkage map chromosomes. So each different line represents a chromosome. And for this, I have the 2 different years of studies showing the size and colors of these small dots are the significant locations, much the same as the genome-wide association studies. So what we find is there are 2 distinct locations that are showing up in multiple years for QTLs in reducing or not reducing inflammation. They're significantly affecting the inflammation. And this is just a blow-up that shows in a little bit more detail the QTLs that we see. And there is quite a lot of resolution here between the QTLs for both years. And the top is chromosome 1, and the bottom graph is chromosome 3. So these are where we got our 4 locations. So now I'm going to try to get this graphic pretty well. Okay, the genotype effect on inflammation. So this is how each one of those significant QTLs is altering the inflammation score that we see. The graphs going down, that's good. That's what we want to see. We want it to be more anti-inflammatory. The ones that are going up, those are not good. The 3 distinct zones are from the parents. You have parent 1 and parent 2, as well as a mix between parent 1 and 2. These are the 3 possibilities that you get in a genomic study such as this. And what we see is that with parent 1, the alleles at QTL 1, 2, and 3 are very strong at reducing inflammation. Now, if you were to get parent 2 only parent 2 at those 3 alleles, then you're expected to have more of or quite a worse anti-inflammatory broccoli. But for QTL4, what we see is that the effect is much stronger, reducing inflammation from parent 2. So from this, what we would be looking for breeders' best broccoli would be AAB, or 3 A's and a B, sorry. So continuing the project, we want to refine our QTL locations that we've already been able to identify some of the candidate genes and their respective metabolites, as well as run similar studies with anti-cancer properties of broccoli. Now this is, as I stated earlier, one of the main features of broccoli is that it reduces cancer incidence. And one of the ways that it can do that is by reducing the proliferative ability of these cancer cells. So we've actually been doing that. That's what we focused most of this summer on. And special thanks to all my interns who've been doing all this lab work for me over the past summer. So what we find is that broccolis with no treatment, and so this is our control, are 100% viable. And this is our baseline, what we find. Then we compare it to an anti-cancer drug called paclitaxel, which is known to destroy some of these cancer cells. So what we see is about a 30-40% decrease at this with this treatment. Now, when we look at broccoli extracts, and this is over the course of that entire population, so these are not all the same extracts, what we see is about a 30 and sometimes even as strong as the anti-cancer drug effect. These are both hugely significant compared to the no treatment. And one of the interesting things we've been able to do with this population is look at the metabolite characteristics that already exist. So we have HPLC measured metabolites for glucosinolates, which have been found to be one of the main anti-cancer compounds in broccoli. They're consistently studied for their abilities. Now, what you see here is on the y-axis is the percentage of cells killed. So this is the ability to kill cells that each of these broccoli extracts was able to do. And on the y-axis, on the x-axis is the amount of glucosinolates. And there is a clear relationship, even though there are a few of those outliers, there is a clear relationship, even so, showing that as you increase the amount of glucosinolates, there is an increase in the number of cells killed. So, going forward, I wanted to end with a little quote. Make sure you eat all your broccoli, said every parent ever. And once again, I wanted to thank everybody that's been able to help me with this project, specifically my graduate advisor, Dr. Slavko Kormaninski, and once again, all the interns and everybody that's been able to help. Thank you very much.